So that's what I take it standard. But it is not necessary that everybody should know chemistry. But yeah, their own expertise will be there, which we will be discussing in some of the slides. We will have an instant feedback session on Menti at the end of the slides, not now. So as I already put up in all my slides, because I collect the good materials from online, so it's not that I prepared everything myself. So I want to give the right to use rights to them. I mean, right ownership. And you can see also Archana was showing that there are a lot of multiple resources like IDs. You can use Sublime Text, you can use Jupyter Notebook, you can use PyCharm, or you can use even uh, uh, like the Visual Studio VS Code or Spider. There are multiple IDs are there. It's your choice which you want to use. It's what is your comfortable for you. So don't think that whatever we used you have to use. We use Jupyter Notebook only because we wanted to have a very presentation mode, and the letters should be much clear and crisp so that you can view it even though the network is bad and it does not have HD. But for the other all IDs, the letters will be small. Of course, you can increase it, but not the way we wanted to present. So that's why we use Notebook. And we don't have any conflict or with any connection with employer-employee. And all the slides will be shared via SlideShare. Okay, and the codes also will be shared by GitHub and Colab. Please do not copy paste the same thing and do train others or do it for your research work. The reason is, then what is your innovation? What is that you're customizing? You don't learn anything. So which I will be covering some of the things. So we also, when we learned, of course, we adapted from others, and then we learned and we customized ourselves. That is what we have to do. And that's only we can learn things. So recently, there are a lot of CADD service market reports are coming. Only problem, to get that full report, you have to pay more than $3,000. Of course, we need not to do that. I got some sample data, which is again anonymized from the web, which I'll be using it today. So AI is clearly stimulating the drug discovery market and providing new opportunities for companies using such technologies. The importance of machine learning technologies is likely to increase in the coming years. This is what I want to, especially, I can see there are a lot of students from pharmacy colleges, IITs, Manipal, and many more, even companies also I saw. So that is good that you have to look forward and you get ready for tomorrow. Recent Times of India survey says that very less percentage of students are ready for tomorrow's technology. So it's the right time, be ready for it. And don't think that simply knowing Python is going to get you a data science job. No, as I told you from my first presentation, statistics. Without knowing statistics, without knowing learning algorithms, no use of Python, right? So don't think that simply knowing coding will get you machine learning, and simply knowing machine learning and statistics will reach you to a job. You need to know also to customization. There are a lot more drug discovery companies worldwide. There are software providers, service providers. So for example, Nairo Research is again a service provider. It's like a contract research organization where we do have projects from pharma industry as well as from hospitals. Now we don't have much projects from India, but mostly coming from US and Europe. Of course, it's not for publication. It is for production and patenting. So we act them as a service provider to them. And we do have custom built pipelines in KNIME, and many of them are already using it. So they tell us that they have specific requirement, what we have to do, what they require, and we build up the pipelines and the workflows. We test it, and they also test it, and they get it. Of course, we get the money for the creativity we did, but the technology will be used by them. Yes. We will be publishing some of the workflows for academia so that they can also start exploring it. We'll be soon doing the launch, and it will be absolutely free of cost, and only for academia, not for any others. Others, they have to buy for those platforms. Now, when you look at the cost analysis, you can see what they're saying, what they're looking is how far it is increasing, they're not looking. How far they can save the cost, that is what they're looking. By implementing what kind of technologies they can reduce the cost. So one of the US-based AI company was telling almost 30% of the total cost and time invested in developing a new drug can be saved by CAD services, including new technologies. That is what they're looking forward to. And it is also said in the recent review, not review, publication, I'm talking about market analysis. There will be an increase of 4.5 billion US dollars by 2025 in the drug discovery market. So why I want to tell you all these things? Be serious. So especially students, you're getting into an era where a lot of technology bombardment is going to happen. So get ready for that. Upcoming trends, artificial intelligence. Now, during this 2 day sessions, we mostly discussed about statistics, Python, and a bit of machine learning. We did not touch deep learning. Of course, we will not touch. Deep learning is something much more advanced. Until unless you're a master in Python programming, as well as you're a master in machine learning, deep learning will be a bit difficult because I told you it's a black box. And you can see the ease of implementation is also not that great because it has its own difficulties. And it's a short term, and the long term, of course, it will be very helpful. Because the problem is we don't have data. We need a huge amount of data to do deep learning. Machine learning itself, we don't have data, but at least we are okay with small amount of data what we have. And cloud-based technology platforms are also increasing like anything. Today, Archana showed her project which she did on autism. So she put all the codes on GitHub, and if you want to just run the code, she has already done it on MyBinder, but she deployed it as a web cloud-based app in Heroku. That's what she said, deployed in Heroku. So anybody can access that link and they can make use of prediction. That means the backend model will be used to predict your data. So machine learning will be used on the backend and it will be predicting the data for you for your set of parameters that you're giving. But you don't know whether it is machine learning in the backend or statistics. It is all on the backend side. On the front end, you see a dashboard, and that is cloud-based technology platform. That is called dashboard. So that is a very well known there, but this is what we were talking about deployments, right? So we need to know how the models can be used by others. That is called deployment. So we are working on QSR deployments. Parshuram sir already knows that we are also working on a mobile app where you have to give the molecular properties, certain molecular properties for specific biological activity or physical or chemical properties, and the model will be able to predict the values. And it will also tell you what is accuracy. Simply predicting value, as I told you yesterday during my presentation, you need to know the standard deviation. That is very important. So technically, the model will be predicting that, that is for sure. And there will be other models also deployed on online, and we are testing, aggressively testing it, but it will take some time for the release. Yeah, but completely it will be a machine learning based models. If you see the distribution, if you can see in the very recent years, like after 2015, more than 25% of the drug discovery company providers have been evolved, and 22% was during almost like 2006 to 2010. So that's where you can see again now from 2020 you can see a bit more because of this pandemic situation. There are many people got into this area. So you are in the hot area time. So it's better that you find out or upskill yourself to the best opportunity. Now, also there is a question whether it is a ligand based or structure based or fragment based. You can see structure based has a more market share with different companies. You can see Jubilant Biosys is an Indian origin company. Then you have AMRI. Again, it has its Indian roots. Then there is Sai Life Sciences. Again, Indian roots are there. I mean, they also have branches here. And then we have ligand-based drug design and fragment-based drug design is also there. So structure-based has its own importance in the CAD approaches. And when you also look at, just a minute, I don't know whether I'm disconnected. No, I'm not. So when you also look at what kind of analysis, these are the expectations by the market. Lead optimization, around 111 expected providers are there. What do you mean by lead optimization? You wanted to optimize your structure. You wanted to make modifications so that you try to satisfy all the drug discovery objectives, basically activity, toxicity, solubility, permeability, metabolic stability, and many more, absorption, others. So in order to optimize it, lead optimization is one of the biggest market. Now, in order to do lead optimization, you should know chemistry, especially organic chemistry with combination of medicinal chemistry. Very much important. Now, heat to lead. Heat to lead is also another criteria where we do a lot of what to say, after doing docking, and then we look at ADME properties and pharmacokinetics and to understand. Don't think that simply screening a database is going to earn you money. No, you need innovation. You need to come up with novel molecules. So simply screening a database and publishing is good for publication, but not for money making. Money making is at least patenting. So patenting means you have to make new molecules. So hit to lead and lead optimization is not that much happening much in most of among us because of the cost, because of the time, and all other things. But we are doing a lot of hit generation. We do a lot of virtual screening. We do a lot of docking studies. We do a lot of bioassay studies. But what is next? We have to reach very close to the drug molecule. So hit generation. So those lead optimization, hit to lead, hit generation is having a good contribution to the market share. And then we have target validation and target identification. Target identification itself has its own market share, which I'll be showing you some examples where it has its proven success in network pharmacology, then systems biology, where to identify the right targets as well as network, the interacting partners, which could be right for your molecule. Now we have to know what is the cause of the failure. There are a lot of failures happening in drug discovery, many, and a lot of market share is getting down only because of the failures. Now, in early, between 1960s and 1980s, these are the two papers I've used to get this data. You can see pharmacokinetics and ADME properties have a major role in the failures of drugs, including efficacy, but not on strategy, toxicity, or adverse effect, because we were always playing around with the standards, which is already being known, and also from the humans' expertise, we were making compounds. We did not explode our knowledge with a lot of diverse space or unknown spaces. We were always focused within our space and with is known to us, which is expert, and so it works. Only problem was ADME properties and efficacy. But in later 2000, you can go through these two papers, it is found that we were able to overcome PK-ADME property issues and efficacy issues because we had a lot of structure-activity relationship models came up using statistics as well as machine learning models, and we were able to overcome that challenge. So estimation of ADME at earlier stages could be done, and also we were able to generate a lot of models to predict ADME PK properties. But we generated another big issue. Because our space got diversified, we started exploring unexplored chemotypes, and there it is more prone to toxicity. And also our strategy, because since we were being also exposed to a lot of approaches and filters and terminologies, conditions, criteria, constraints, we simply applied everywhere. Wherever others applied, we also applied. So strategy became a bit wrong. Some start with SBDD, some start with LBDD, mix it up, and we don't know where it is going. So strategy and the toxicity has come as a bigger major cause for the cause of failure in the current trend. Now, this is where we have to focus on. That's why recently you can see a lot of machine learning applications on retrosynthetic pathways to design new molecules, whether it is synthesizable or not. What is a knowledge-based method for toxicity? Or can we design new molecule avoiding toxicity? So all those questions came up. But still there is a question, is machine learning is overhyped? Because chemists weigh in the techniques, possibilities, and its pitfalls. And this was published in CNDN during 2018. Of course, already 2 years passed, but still it's worth. So machine learning is totally overhyped. machine learning is completely not overhyped, but it is transformative. And it is also a tool like any other. You have to use it in the right way. As I said in the first presentation, don't think that simply machine learning and all these statistics giving a result outcome, it's always right. So that's a problem here. So you have to make sure whether the data has its consistency. So they took around 151 survey respondents, and they found around 31% agrees that ML is overhyped in drug discovery or even in chemistry. But it is now overcoming. As I said, it is also not overhyped. Has to be put in the right place. So that's why I am telling everybody here, please don't think that, oh, I don't know machine learning, others are knowing, am I going to be behind ages? No, never. But if you start in the right way. I get a lot of interactions from students that they wanted to do internship. Of course, our internships are not paid. I mean, we don't charge them, but if they work for our project, we pay them. But we don't simply take an intern. We want to know whether they have basics of statistics. We want to know they have a basic understanding of scripting. We don't want them to know Python completely. We are here to teach them. But basics is very important. So that's what I'm trying to say. So just attending a conference or a workshop for 2 days, don't say that you know machine learning. If you yourself need to know that, confirm that you know machine learning, take up a project or a problem from somewhere, or there is a website where you can take competitions. Try to win at least 2 or 3 competitions. I didn't say prize, but try to solve those problems. If you are able to solve it by yourself without copy pasting, but thinking yourself, and of course, all the errors you have to search anyway, then you put in your CV that you know machine learning. So that's where it is overhyped, because many non-experts claim that they know machine learning. I will never say I'm a machine learning expert. I will say I'm a beginner to machine learning. That's all I will say, because I'm yet to explore and still learning every day. So it's about your choice what you wanted to do. It's about completely your choice. So here you can see about 151, 23% were organic chemist, but only 7.3% were computational chemists. And it is understandable, because computational chemists already have tools to predict their whatever properties they're looking at. But we are still also working on that. Some of the methods will take hours to finish or days to finish. But using machine learning models, we are trying to see whether we can speed up the calculation time. Even the impact of artificial intelligence in CAD. As I told you, we are nowhere in AI in drug discovery anyway, but for the media, they rather than ML, they prefer to hear it as machine learning. Sorry, artificial intelligence. But of course, deep learning is being applied in many places where deep learning and artificial intelligence coming, and if there is an automation coming in, automatically you can call it as AI anyway. So there is an exploring chemical space. Can AI take us where no human can go before? Absolutely yes. So already we have enemy in databases, which has more than 3.4 billion database compounds can be generated. And what is their technology is that you can build up or you can build a new compound on the fly using those building blocks. So that building blocks will help you to generate those 4.3 billion compounds. That means don't imagine they already synthesized 4.3 billion compounds. No, they only have the building blocks with them, or intermediates or starting agents with them. But with that, the machine learning models or a deep learning models were able to generate that particular data is able to generate 4.3 billion compounds, and with an accuracy of 85 to 90%. That is absolutely wonderful. 85% of accuracy on this 4.3 billion is still great. Merck has already tried, and there is already a deployed software available called as Infinicy. Infinicy does this to explore the chemical space. So that is the technology. Now coming to AI identifies drug candidate in weeks. Yes, within 46 days, I think they were able to identify a compound. This is again another AI company did that. They came up with a potential kinase inhibitor, right? And they also published that in Nature Biotechnology. But now, whether this is successful or not, we have to wait and see. Now, let us see how these machine learning workflows work in chemistry, right? Because I'm sure many of the chemists are here, pharmaceutical chemists or others, even pharmacologists might be there. I'm coming to you soon, but still, let's start with this. Your main work will be synthesizing compounds and other things, but still you want to look at the yield, which reactions were successful, which reactions failed, all those things. So you wanted to annotate the data with production. And if you're working on the materials or polymers, you also look at sensors, screening, synthesis, simulation, etc. Many data for that single compound. Now you have to do pre-processing, cleaning, integration, transformation. I hope Archana covered that while she was talking about data frames in pandas. So you have to do transformations, you have to do splitting, missing data has to be handled, all those things to be taken care. Then we apply multiple models to come up which could be the best model. So today she was also talking about autism, where she applied SVM, classifier methods, then logistic regression. She also tried naive Bayesian method, and then I think she showed you the accuracy levels of each different models, and then we make a conclusion which one will be the best. And that's where we do training. So we try to learn, make the model to learn which one could be the best, and then we can come up bring with the new data to train them. So ultimately, whatever the model, whatever she did today, it is not taking the data from the user, but there is an option when we make a dashboard, when a user submits the data. For example, we made a model, we made an app, and the users can predict the whether the child is autistic or not by giving their personal data. There is a provision that we can also accept that data and improve the model, improve the accuracy model. That is also possible. But we have not done that because we don't want to take any personal data as of now. That is the reason. So new data also can be put in, again, retrain the model to improve its accuracy is also possible. And mainly it's based upon the data types. I told you in the first session, data types of quantitative and qualitative. It's a regression or classification based. Now, how the chemistry data is being used in machine learning. Let us see the first aspects. I'll go to the coding and all those things. Don't worry, I'll be covering that today also. But I want to make you understand where it is applied. Maybe you would have read already, but I don't know how many of you have read. That's the question. So project-oriented data sets, fundamentals of working with active learning algorithm. This is what example that I right now said. It can take data from your existing real data, and the model does active learning and improves it by optimization. So that is also possible in your machine learning. So it's not simply that you had your initial data which you started with training the models, but you can also get data while other experiments are going on and the model gets learned by itself. So this will help you to minimize the number of experiments because you might have already carried out or it is similar. Machine learning is able to overcome that, so save money. Second, you can maximize the materials properties because since you already know this, already a model is already available which could predict something with better accuracy. So let me look at or concentrate on improvising the properties by making modifications on the structures. Another example is framework for working with in-house databases. I'm not sure how many of you are maintaining in-house database. When I was working with Professor Alan Kettersky at University of Florida, he had an in-house database. For example, Sigma Aldrich used to buy compounds from my professor. That's the case. We were having a good tie-up with Sigma Aldrich. So he used to synthesize. We had a group members of around 48 members and a lab around 7 labs, full-fledged labs, completely for synthesis, heterocyclic, macro, then peptidomimetics and others. So I was on the computational group, but on my curiosity, he permitted me to learn synthesis also. Of course, I'm not a master there, but still able to read NMR and other things. Anyway, why I said that is, when you have an in-house database, you come to know about your reactions, historical reactions, which were successful and which were failure. Normally, in what is our scenario, we never publish what is failed. We never publish what is negative, but it is required. Let others know it. That is where the importance of machine learning comes into the picture. So once you get to know about the failed data and success data, we can generate SVM models because it's a categorical, right? As I told you yesterday, yes or no, true or false, win or fail, like that. So we can go to SVM models and we could look at different various reactant combinations and the constructions can be built up and then we can further test, optimize it, and then the model will be able to tell you whether this reaction is going to be successful or failure. So that is the level. It is really working actually. But the problem is you need to have data. How much in-house data you have. Other in-house data, they will not share it with you. So don't try for that. If it is they are sharing, you have to pay. That is the reason why you are paying for SciFinder. SciFinder curated all this data and now you have to pay for that. Without that, you can't get access, right? So that is the reason. Another one is data from simulations. I told you earlier, there are DFT calculations and some extensive MD simulations, which could be, or force field parameterization, which could be overcome by ML. So those are also possible. Yeah, those are also possible. Here you can see by using machine learning, you were able to predict the energy in 3.13 electron volt. But DFT, it gave you 3.17. Absolutely fine. I think if it is the time we can overcome much faster, the compromising the accuracy, it's absolutely fine. That is anyway well known in DFT. The level of theory and a time taken for convergence. But when you're working on public database, you can use neural network to predict the reaction condition. So neural network, CNN, ANN, and many more other hidden Markov, all these models goes bit advanced. So until unless you know whatever we are discussing these 2 days, much more in a very legible way, we cannot go or touch base on those areas. Now let me explain what is this project is about. They made the reaction. There are plenty of reactions came in and they put it in the neural network. That's here, it's a black box. We don't know what is happening in the hidden layer at the middle. We just have the input layer and the output layer, but in between what is happening, we really don't know. What they have done is, by the model, it will be able to predict you what could be the best combination of catalyst, solvent, reagent, and temperature to get you the best yield or purity. So there are neural network models. So don't try that you are getting a free code online. No, these are all mostly somebody tries to make money out of it because it is their innovation, right? So keep those things in mind. You don't get everything available for free online. Now, deep learning applications are also there apart from machine learning. So DL algorithms for solving different chemical challenges and also the respective task. One method is designing new molecule. Now, how do we design? You have to understand the molecule based on the property. So based on the property, you can design the molecule, or based on the transition state, or what could be the product and what could be the reactant, you can predict the imaginary or images, intermediary images. That is also possible, even without DFT calculation. Synthesizing molecules. What are the possible routes we can synthesize the molecule, in which we can predict reaction prediction, retrosynthesis, and reaction optimization. Coming to understanding the molecule, you can accelerate to quantum mechanical calculation, QSPR, and conformational exploration. Because conformational exploration is also important, because if you wanted to synthesize and crystallize the compound before crystallization, if you can predict them, that is always good. So whatever data you have, you need to have a very right data, and we will put it into the learner, we optimize it, we have a training cycle to predict, evaluate, and then optimize, and then finally come up with a predicted model, right? And then for data analysis, I'm sure Archana would have covered matplotlib and pyplot, which is again within matplotlib. There are several representations available like box plot, a violin plot, then histograms, scatter plots, heat map, pair plots, many more are available. Each one has its own importance and all cannot be used in all kinds of data. So you have to know the representation for which kinds of components. The prediction performance, anyway, Archana already showed her with her project how different models are giving the prediction performance. This is also from Nathan, one of the very big pioneers in AI in drug discovery. So they have published taking the public data from Kager, then NIH Tox 21 data, and they were able to predict what could be the accuracy value, that is AUC values. For numerical data, of course, you have R square. And you can see the R square, they got 0.49 and 0.46. Did they get 0.81? No. So that is what we are looking at. How is the performance of the model coming in? Now, do you give a 0 mark to this particular model? For the first one, 0.49 R square? You cannot, because it's completely dependent upon the data. And you have to understand what is the other relevant parameters to justify what is the performance of the model. Now, how do we convert these structures to machine learning understanding language? So this particular structure is very easy for us. We know to understand what is the angle, what is dihedrals, what is bond length, bond angle, what is the element, everything is fine. But the problem, the computer does not understand. So in order to understand, we have to convert this data to something computer readable. That's where in machine learning, everywhere you hear something called as fingerprints. Without fingerprints conversion, machine learning algorithms cannot take your data directly. And we do have algorithms, libraries for that, like RDKit, CDK, and many more other fingerprint, like Maybridge algorithms. Many are there available for you to convert it. So the ideal features should be general, compact, unique, invariant, smooth, and fast. But issues with machine learning is arbitrary size and arbitrary order. Now let's see how the whole molecule is given to as a global descriptor, but how localized or atomic descriptor, because for a whole molecule there is a particular fingerprint, but for the each regions, let it be an atom or a bond or a region of the compound, they also have respective fingerprints. And these fingerprints will be unique. That is important, right? Now let us go to some of the demo. Now I will use KNIME. It's a live demo because I understood that even though machine learning without Python, machine learning is nothing, because machine learning is for Python is anyway required for you to do any customizations. But if you're not okay, if you're fine with whatever data is there, and you try to do pre-processing of the data and you come up with whatever possible, then you can use these KNIME and Orange. So don't worry, people who think that, oh, Python, lot of things to learn. I don't have that much patience. I don't want to do that because that is not my domain that I'm looking forward to. You can use KNIME and others, because that is anyway completely fine with you to kickstart with learning with Python or ML also, because ML mostly you are looking at application. So this is absolutely fine. You can use KNIME or others to start with your application. Now let us go through some of the selected drug discovery nodes. So you have something called as 3D EChem, Alva Science is paid, then CDK is there, NLOs, Earlwood, Indigo, Lhasa Limited. If you want to do some next gen sequencing, RDKit, Vernalis, these are the selected drug discovery nodes available. And some of the commercial drug discovery nodes are Chemaxon. Of course, Chemaxon has its Marvin Sketch, Marvin View free of cost, but others like JChem, cartridges, they're paid. Biosolve IT, some of the nodes are free, but for docking, virtual screening, chemical space exploration, it's paid. For Crescent, all the nodes are paid. Molsoft, all the nodes are paid. For Schrodinger, some of the 2D to 3D converters are free, but most of them are paid. You have to pay. And MOU is also paid. So these are all commercial nodes. The one which is colored, they are all open source nodes. Now, some of the nodes also has the specific extensions as some functionality. For example, Vernalis, you can do confirmation, generation of confirmation using force field and RMSD. Principal component of inertia. You can generate a fingerprint. You can access PDB databases. You can also have matched molecular pairs. And yeah, you can retrieve PDB based on certain criteria. And you can also search sequences and do sequence compare analysis. Earlwood, again, you have data converters, for example, something similar to OpenBabel. So there is OpenBabel node also for KNIME. So you can do conversion of multiple compounds from 2D to 3D or from SDF to mold, mold to mold 2, all those things can be done. And here also in Earlwood, we have virtual screen. Don't think this is docking, but it's something entirely different. But we do have docking within KNIME, which uses eCheminfo. I'll show you in a moment. And then we have similarity searches. If you want to search a compound within the same database or other database, or if you want to do a database comparison, those things can be done. RDKit is something very famous in Python because they have a Python wrapper for RDKit. But otherwise, they also do generate a lot of cleaning of your structures, then descriptor calculation of molecular property calculations, then geometry optimization from 2D to 3D, or a normal molecular mechanics based optimization, Marcus enumeration, reaction sampling, and many more they also have. And then CDK also mainly, earlier days it was mainly used for prediction of X log P, but we also have cleaning of structures, Lipinski rule of 5, and extraction of data from ChemSpider also could be done. And some of the selected commercial extensions are Biosolvite, Chemaxon, Crescent, and MOEN, Schrodinger, and many more others are there. Of course, I don't have access to any of them, so I'll be not showing any of these modules. And of course, you have to pay for this, so there's no point if I show any of them. But otherwise, all the other earlier showed ones, of course, it will work. Let me also now show you one of the some of the key nodes in KNIME, which will be useful for basic chemistry. So for example, the first one, Chem Sketcher, Marvin Sketch, Structure Sketcher. These all is going to help you to draw structures. Whatever you do mostly in ChemDraw can be done here. Of course, drawing a reaction is not always our job here anyway, but still you can do it in Marvin Sketch. Marvin Sketch allows you to draw the reactions. So for drawing 2D structures, these are the drawing tools available. Then we have converters or not converters, reading the files. You can read any type of structure files, SDF, mol2, mol, PDB, all those files. Smiles also. This will read SDF files, mol2, mol file, SDF, Smiles direct to read reactions reader. So if you have any ChemDraw drawn reactions, of course, R group analysis, Marcus enumeration, those things can be done. Then we have converters. Some softwares require specific file formats, otherwise it will not work. So you can use mol to CDK, RDKit from molecule, OpenBabel, and mol converters from Chemaxon. Then we have visualizers. Marvin View will help you to visualize all the 2D, 3D structures. Ligands Viewer is a 3D viewer which helps you to visualize the molecules. Pharmacophore Viewer, which will help you to view the pharmacophore mapping. Proteins Viewer will help you to view proteins. Ligand and Protein Viewer will help you to look at the complexes. So all these basic nodes are also available in KNIME. Then let us look at the chemistry calculators. Sorry. So we have fingerprint generators, for example, where is it? Yeah, this one. RDKit are molecular property converters. They generate fingerprints and then only they'll calculate the descriptors. And you can also predict descriptors and molecular properties. Keep in mind, most of the nodes might predict similar descriptors. So when you are combining them, try to remove duplicates. Otherwise, you end up with lots of overfitting issues. PADL is again available in KNIME, which can generate descriptors and molecular properties from Indigo, molecular properties from RDKit, then Enilos, all those things can be done, including PDB property extractors available. This, I will be showing you a demo today where you can predict all the developmental toxicity using KNIME. Then you have output port or table. For example, you can write mol2 files, mol file, SDF file, Smiles directory, CSV file, Excel file. You can even write pharmacophores as well as table writer. All these are readable by other softwares, any software. Now, when you look at the table, this is called the row ID, and this is the column header. So you will have the heading title. The integer data type, it will tell you whether it is an integer or a float or a string. If it is written in S, it tells you string data type. So keep in mind, KNIME is mainly used for data analytics. So it will also try to tell you more information about your data. So it is telling you what is the kind of data type, whether it is a string, integer, or a float or object. Yesterday, Archana was telling you about data types in Python. Now, this is one of the workflow where you can use it for converting your 2D chemical structures to 3D and including optimization. All these I have, this I have posted in GitHub. So you have this in GitHub. All the instructions are available. So I have created this in 2018. So almost 2.5 years old, but still works. So you can read a mol file or an SDF file. Your job is to just connect to the right one. Either you can connect from mol to RDKit or SDF to RDKit. Both will work. Only I made it did not connect because you have to, I don't know which file you have. So you can read either mol or SDF. And then use RDKit from molecule. And now you are adding hydrogen. Even if you have or not, it will add hydrogens. It will do geometry optimization, and you can view the data with the energy values, which can be saved as a CSV file. Another criteria is you have the 3D coordinates already with you, and we make it CD2 molecule. So CDK or CDK node will not the scenario. So sorry, again 2D molecules you have. CDK will use 2D to 3D converter, and it will, you can write it as a SDF writer, and also you can view those 3D. These two nodes can be also used here, as you see here. Multiple things can be written. So from here, I am writing it as a CSV file. Also, I can write it as an SDF or as a 3D view. All are possible. Now let us look at some environmental toxicity models. Similarly, mol2 file, SDF file, or mol file, anything can be bring in here. And but here the question is that we have to convert it into smiles. So converting to smile is mandatory, and then we take them to Vega models. Unfortunately, Vega does not have extensions right now available in the repository. So I'm not sure whether it might work for you. I am having an older version, so it still works. Now, if you ask me to share those files, that node will not work with the latest version of KNIME. So that is also a problem. I told you, 2.5 years back, and I was using KNIME 3.5 version. Now the KNIME version is 4.2, so you can see the difference, right? And then we try to merge all the data and write it as a CSV file. So for all the chemical structures, we get the mutagenicity, carcinogenicity, developmental toxicity, skin sensitization, fish acute toxicity, and A log P. Now, this is another workflow where you can use it for generating confirmation. Don't worry, it's not only slides. I'll be showing you a demo also. I think we still have time. Yes, we do have time. So generating confirmation. So I can draw a structure or I can load a structure. Anything is possible. So here I am drawing the structure, and it is converting that structure to RDKit because it reads fingerprints. And then I'm using RDKit to generate confirmations. And for each confirmation, it is calculating the energies. Of course, energies to be there. Redundancy has to be removed. Duplicates to be removed. And we can also do a template conformer generation. Means we have a reference conformer, or we set the template, and within that particular space, it generates a confirmation. Energy-based confirmation analysis uses universal force field, MMF 94 force fields also. Then if you want to predict which site of metabolism, let's say it is not about predicting metabolites, it is not about predicting metabolic stability. It is just telling you how far the energy, accessibility, and solvent access area of your drug molecule for the P450 isoforms 3A4, 2D6, and 2C9. So it tells you in the quantifies in energy. Its energy is an approximation of activation energy for the reaction of the catalytic site of the CYP cytochrome with the molecule of at this particular atom. So this will show you that information. The accessibility is a relative measure of topological distance for an atom from the center of the molecule and is always a number between 0.5 and 1. Solvent accessible area is about describes the local accessibility of an atom and computed using this particular algorithm, which predicts its value with molecular topology. Again, you read a molecule, RDKit reads, converts it to fingerprints. We generate the 3D coordinates, and then SmartSIP tells you these values, and which SIP tells you your compound might get metabolized or might get more attractive to either to 3A4 or 2D6 or 2C9. It just tells you which one. That's it. So there's a paper published in which SIP. You can read the paper to understand a bit more. Another thing is that let's imagine that you have an in-house database and you're doing a very secret project, not secret, confidential, which you're planning to synthesize or some patent or something. And you don't want to upload these files to PubChem. So don't do this hereafter after this seminar. Because if you upload a novel molecule to PubChem, after 4-5 weeks, you will see that compound will be listed there. Of course, there's no name. They will clearly mention someone has searched it. So if you're thinking to patent some molecule, don't search in PubChem. It will be public. So in that case, if you want to do this searching or locally, what we do is we download the whole database, okay? Then we draw a structure and then do a comparison to find out the similarity search. This can be done offline. Nowhere it is going online, nobody stores your data. So for that, we can do a substructure and a similarity search. In the SDF reader, I will be loading all the database which I have downloaded. Keep in mind, if you are reading a large data set of 1 million or 100,000 molecule, keep in mind you have a good RAM for your computer. So whenever you're doing anything for data analytics for large data set, you need good amount of RAM actually. Memory is more required. CPU and GPU is required, but that is for specific cases. For reading molecule, writing file and reading large files, you require a good amount of RAM. So keep in mind when you're upgrading your machine, you have a good amount of RAM and good amount of cache for your processor. Because even though you see i5, some i5 will be cheaper, some i5 will be expensive. Why some i5 is expensive? Because their cache size is larger than the cheaper one. So I have a machine with cache about 32 MB, but normal i5 machines cache will be of 2 MB or 4 MB, maximum 6 MB. So most of the time we don't look at all these things, but yeah, since we are working on data science and large data, we have to look at it. Otherwise tomorrow you might think I bought an expensive machine, but still it's slow. It's because of many minute things. So don't think that your gigahertz speed in processor, it's going to help you read things much faster. You know, it will process things faster, but for reading you require good RAM and cache. That is important. Anyway, if you want to know more about it, I have a video in my YouTube where I built my machine. I discussed about all these small factors. Anyway, so you have to load your PDB database here in SDF reader, and Marvin sketch is where you draw your compounds which you wanted to search. So you have to convert all the database molecules to fingerprint and your molecules also to fingerprint. Now we are doing a fingerprint similarity search, and then we generate a distance matrix and similarity here. So we get a heat map, and we can also compare from one compound to the other compound, even a compound within the database, all things can be compared. So we get a distance matrix definition. Now, we can also do pharmacophore screening. So you have a data set with you, and you have your, reference molecule. So a reference molecule need not to be drawn. If you're doing 2D based, then Marvin sketch is fine. But if you're doing a 3D based pharmacophore, keep in mind, you have to bring in an X-ray crystal structure 3D conformation or a bioactive conformation. Bioactive conformation is 100% preferred, right? So don't do a 2D to 3D conversion to make your mapping or generating the pharmacophore maps. So if you do not have, then use a 2D, absolutely fine. So it is more of a field topology based. But 3D, consider your reference molecule to be always your 3D bioactive conformation. And then your database will be in the SDF reader. You can generate the pharmacophore for your scaffold, so for your bioactive conformer. And also you can generate the pharmacophore for all your database molecules and then connect it for the aligner. So the aligner node will try to compare a similarity between the pharmacophore of your reference molecule with your dataset, and it will generate the pharmacophore viewer. Of course, I have to connect this to get the viewer. So this is possible. Another thing is, if someone is working on QSAR and you want to generate molecular properties or descriptors, we have plenty of descriptor property predictors. I'll be showing you. So you have molecular property predictors from Indigo, XLOCP from CDK, RDKit descriptor calculation, then Enilose, then Enilose Mol2. Of course, Enilose Mol2 will work only for Windows, but not for Linux or Mac. It will work only for Windows. And then you have pedal descriptors, and then you can join all of them together by understanding which has duplicates, and then you can finally write them to a table to do machine learning or statistical based learning modeling. And Orange, I won't be showing a demo today because that day I quickly showed you something. So, but we can do something called feature rank. Archana did not show this, and we purposefully avoided it because feature ranking is something that we wanted to find out which is the best variable, or which is the best molecular property, or which is the best features which will be reliable to make a very good reproducible model. That is what I'm trying to say here is, let us say we calculated 100 different molecular descriptors or properties. And when we use 100 properties, your model will give you an R square of 1 or 0.999. Doesn't make sense because it's too much of overfitting, too much of false positives. We do not know whether the molecular properties 1 and 10 is very well correlated. We don't know that. In order to do that analysis, we have to do feature selection or feature ranking. And another thing is, we also wanted to know whether there is any missing data available. This is called the data wrangling, WR, wrangle, right? Data wrangling. So we want to do transformations. We wanted to do modifications. We want to analyze missing data also. So imputing the missing values to able to visualize all the data points also. So imputation is also another challenge in most of the data where we have missing data. So deep learning methods are used in drug discovery to do imputation. So recently there's a software released called Cerella. Cerella is from Stardrop Optibrim. They do deep learning based imputations. I don't know any others doing the imputation, but there are many other imputation techniques available. But bioassay data imputation, I don't know any other tools available. And also we do have to display the feature score to understand which feature is more important and which feature is less important. And we also have to generate a feature map or a heat map to understand which feature is having more relation with the other one. As I told you earlier, the first feature is having around 95% or 85% correlation with the 10th feature. We shouldn't take both of them in a single model. That will create overfitting issues or false positives because the model itself will get confused whether to take the first one priority or to give the 10th one as a priority. So covariance, I discussed about covariance, intercorrelation among the features or descriptors also has to be taken care. And now we have to reduce the data. So reducing the data doesn't mean that you're simply removing all the values. No, you have to remove or reduce or prune or remove the duplicates based upon the feature selection analysis. Another is classification tree. If you have a numerical data, of course, you can always go for regression studies. But let's say you have a categorical data, yes, no, or the plant will survive or plant will die or something like that, categorical data, then we always look at classification tree. And we don't know how far we can predict them, because there are certain algorithms which is specifically for categorical data. And this is how these models are also available in Orange. Orange does not do any chemistry related elucidation. Orange does only data analysis and model generation. Orange has a little bit of functionality in bioinformatics, but otherwise it's completely text mining and data mining, including machine learning, statistics, and generation. But KNIME, as I told you yesterday, it has many more chemistry-based or computer-aided drug designing-based workflows available. Now, finally, cross-validation. Yesterday, I was telling you this. We have to cross-validate it. Cross-validation can be done in several ways. One many out, leave many out, or fold, how many fold you can do. And then you can check the performance of those models. And you can do confusion metrics to understand false positives, false negatives, all those things. And you can calculate the accuracy, error rate, then precision. All those things can be calculated from confusion metrics. And then here also you can see here, they have used logistic regression, random forest classification, SVM, and then we do a cross validation at test and score, and then confusion metrics, right? So these are, Orange completely use it for data what you already have or generated from KNIME or some other softwares or from experimental data. Don't use Orange to generate data. It doesn't generate data. It only analyzes the data. So this is what I was telling you. Whatever materials that we are giving, adapt from that. Don't simply copy paste. If you copy paste, you don't learn it. That is the sad part here. Then if you say, I know things, sorry to say, it will be difficult. So don't feel offended. I'm telling you the right thing. Try yourself by typing, by looking at it, type yourself. Don't copy paste. Type yourself, repeating it again. If there will be a big difference between putting capital letter, small letter. If you miss a space, it has a big difference. If you don't put it parenthesis in the right parenthesis at the right place, it will have issues. There's a big difference between single quotes and double quotes. So when you copy paste, you don't realize that, because most of the time we know that we learn from our mistakes and we remember that. So see it, type yourself if it is a code, whatever it is. And if you made a mistake, that is a human error, right? And then compare whatever even for it worked for us, why it did not work for you, because there are some minor mistakes you have done, and then you analyze it. So you have to learn, but apply with your own ideas and creativity. That is something very important. Otherwise, you will never learn and apply somewhere, because copy pasting will always help you to learn, but you cannot apply it in many places. And until you try yourself and train others, you cannot be an expert. To be honest with you, after my master's, the first job I entered was a lecturer, right, at Salem for bioinformatics, basically. To be honest with you, I learned more than what I was preparing for my examinations in my master's. I learned more before facing the students in the next day of my class. I have to sit, read, prepare the material that I have to teach them for the next day, right? So unless you try yourself and train others, because when you want to train others, you have to be more confident. That's where you have to be very rigorous. Okay, I have to learn, otherwise it will be difficult. I shouldn't tell anything wrong to the students or to the others. You will never be an expert, and that increases your confidence also. That is my take-home message for this all throughout. So thank you so much. And then right now I'll go to a quick demo. Here you have my email address and my contact details. If it is so urgent, you can call me, but otherwise I have WhatsApp in it. You can, or I have WhatsApp, Telegram, Signal in it, because nowadays too many concerns about privacy by many people. I will be still using WhatsApp anyway. So anyway, all are available. So if you have any suggestions or something that is required, you can always contact me. Now, let me quickly go to some examples and then demo. So these are some of the workflow in KNIME for getting data from Kemba. So you can filter the rows and all those things. It is available in publicly. Hope you remember, I requested everyone to sign up for an account in KNIME Hub. This is accessible via KNIME Hub. I will not be giving you. It is in KNIME Hub. So start an account and go there. So you can, this particular workflow can be straightforward downloaded, and you can convert your bioactivity to PIC50. There's a math formula. It's already being used. This is already standard. You can download it and use it straightforward. And then molecular filtering based on ADME and lead likeness, not only based upon Lipinski rule of 5. So here you have again Lipinski rule of 5. And also you can calculate certain properties using RDKit, and then we generate ADME properties and then compare them. And then ligand-based screening. Again, we use RDKit for generating the fingerprints. We do similarity search, and then we do an enrichment plot to see how far the best chemotypes came in, and then we did. So don't think that this is a QSAR or Pharmacophore. It's some completely different types of similarity, structure similarity that is being based upon Morgan fingerprints and MAC fingerprints. There are several types of fingerprints available in RDKit, which I'll show you in a moment. And then you do a compound clustering. So yesterday someone was asking me about the clustering. So you can do, again, based upon a specific fingerprint. So as the fingerprint changing, things are going to change. So keep that in mind. It won't be same. So here the Morgan fingerprints are being done. They did a Tanimoto distance matrix calculations, and then did a hierarchical clustering, and then it could be visualized for the compound clustering. And then a machine learning based ligand based screening is being done. Again, the structure is being, or the smiles is being converted to fingerprints. And then we made a random forest to learn, as well as artificial neural network classifier and SVM model. So we learn and then predict. That's what machine learning does, right? So we learn and then predict and then aggregate, and then we evaluate the model for random forest support vector, and then finally come to know the ROC curve and the score view. So this is complete the evaluation process. Now, how to make use of this model? It's a different step. That is called deployment, which I had already showed you yesterday, but I'll again today focus on it. Yes, this is another project from KNIME again. It is called Open Facts. They do have combinations with a lot of databases like Disginet. Disginet tells you about the relationship between the genes and the diseases. Then Wiki Pathways, then Gene Ontology, Uniprot, Drug Bank, ChemSpider, ChemBell, NextProt, KEBI, Concept Wiki. So OpenFacts does a lot of NLP, language processing, text mining, data mining, and they combine to get you all this data, like physiochemical data, identifiers, and pharmacological data. Earlier days, when I was doing QSR, I have to sit and read papers and draw manually in ChemDraw or Marvin 2D structures and convert it to 3D and then do docking and all. Has to do one by one. Human errors will be there. But right now it is very easy. Just directly go to Campbell. You can categorize the data based on bioactivity or assays or a paper, and click on a button. You can download the SDF file, Smiles files. So right now getting the data is not a big deal, but you should know how to get that. That is the deal here. So that you have to read a lot to understand what kind of data could be accessible. Let me directly go to KNIME now. So I am in KNIME. I have the nodes available here. So let me show you quickly. I don't know how many of you went through the KNIME website, the video which I gave you yesterday in Slack. But let me see how many participants are here. Okay, 21. Okay, still many are missing. I don't know. Okay, no questions as such yet. Okay. So we can read the SDF reader. So let me search SDF reader. So I have SDF reader here, right? I can just click and drag and drop here. That is one option. Now how to install these? Sorry, I have to go to help menu, click on install new software. ensure that you're connected to internet, and then click on this and say all available sites. It will try to load all possible extensions of KNIME. Some are paid, some are free. It's up to you which one you want to install and which not to install. So depending upon the internet speed, it will show up. Okay, it is taking some time. Let me try to show you only one because it is trying to take everything. I'll take this one. Yes, yeah, it loaded. So some of the data, extensions are available. Let me try whether I can get you, community. So here you can see CDK. CDK will be using Earlwood for Cheminformatics. Then NGS if someone is working on sequencing. CDK, LASA, we have many of them, LASA. Then RDKit we have, right? RDKit. Then then others will be, and we have for partner extensions. Yes, so most of them are paid. Chemaxon, only few of them are free. Ligand Scout paid, MOE paid, Schrodinger paid. Yes, so these are all paid. You just click and click next, it will get in. So all the paid ones also you can install, but it will not work unless you give the license. Right, and when you click on a particular node, right, when you click on a particular node, you can see on my right hand side something is changing. So this gives you the readme, so you can clearly understand what is the input and output required. If I click on here at the bottom, it will tell you what is required in your input. Sorry, where is the input? Input resources. So input means if there is no arrow in the other side, that means it is to open a file. If you have an arrow here, that is the input. So inputting structures, and this arrow on the other side is output. But here we have two output. What is that? These two arrows are two outputs. Let's look at it. The zeroth output is table with red molecule. So any molecule that is red and is successful, that is red molecules. The second port, which is called 1. So yesterday you known from Archana that indexing, we always start from 0, not from 1. So here the zeroth port is the read molecules, and the first index port or the port is broken molecules, which did not read. So by going through this, we will get to understand what is the input, what is the expectation, what is the property handling. All those things will be understood from here. If you want to delete, just click the right button and say delete. Now let us see what is this here. Just double click on it or click the right button and say configure. Both goes to the same window. I already have a data set already opened. You can browse and open a SDF file. Property handling, you can scan or extract all properties or scan properties. Now I can see there are 191 molecules in this data set. I only have 3 columns: compound ID, NK2 pKi values, and the series. A pKi values is a number, compound ID is an integer 1, 2, 3, and series is a string, some text, right? And then, okay, you have to click. I'm clicking X because I have already run this. I don't want to rerun it because it takes some time. Now, Marvin sketch. Let us search here to work with Marvin. You should have Chemaxon to be installed. So all this under Marvin is free. All thing under JChem is paid. So unless you do have license, JChem will not work. So Marvin, I just drag and drop here. I double click and you will see the drawing tool here. So I can draw tools here. So I can make modifications. If you want to bring in your periodic table and add, you can do. So anyone who is using Marvin, it works. All things work. You can even open files without drawing. You can directly open files also. You can copy paste it. You can copy the smiles and paste it here. It will automatically draw. All things are available within this Marvin. I generated fingerprints. Let's see indigo fingerprints. So here I have molecules and it went with fingerprints. What are the different types of fingerprints? Similarity fingerprint, substructure fingerprint, resonance, tautomer, and full fingerprints. I'll show you a few more fingerprints in RDKit anyway. And then we are finding the fingerprint similarity. I'm telling which is my query molecule, which is my reference molecule, and what kind of metrics I have to use, Tanimoto or Euclidean or Tversky. you have to understand what is the importance of each one of them. I'm not going to explain here anyway. It takes more time to talk about all these things. Mostly Tanimort and Euclidean is what we use. And then we generate the distance matrix. When I just double click, it will show me which one I have to consider for generating distance matrix. And similarity viewer, again, it's asked me which is a structure column and which is the distance matrix. But in order to know or see this, I all discussed about the input variable and other. If I want to connect from one particular thing to the other, so let's say I have a Marvin sketch and I have a viewer. Okay, I have to connect this to this. First, this will finish this process, then only this process will. So I have to click here, and then you can see there's a connector coming, and I click and connect it. So I connected it. There is a traffic like signal like of things. Here this will be red, yellow, and green. If you want to see red once again, let me do that. I'll just delete this. And now you can see there's a red. Red means it does not have sufficient data to start. Yellow means all sufficient data is ready, it has to start. When it comes green, it is completed and ready with output. So I'm just connecting this. I go to this and I'll draw some structures. Okay. And adding something, double bond, oxygen, and then carboxylic acid. And I click OK. And view, I want to view the structure, automatically the thing I want to do. If you want to run a single node, either you can click the right button and say execute, or click that particular node and click on the top, the single play button. If you want to execute only specific set of nodes, you can select them and say execute. Without selection, if you want to execute all of them, whatever is there in the workflow, you can click this particular button. As you can see, execute all executable nodes. Keep in mind, it will do only one by one. Where you started, first it will complete this. The output of this will be used as an input for this one, and the output of this particular node will be used as an input for this one, and then it gives the output. So each one, it goes one by one. If you're doing for a larger data set, I recommend start with a small data set, test your workflow first, and then only start with a larger data set. Otherwise, you're wasting your time, you'll be having trouble. Now let us execute this one. So I selected it and played. So this is run and this is also run. Now how to view it? Click the right button and say view Marvin view. Otherwise at the bottom there will be some other data. So view Marvin view. I have the structure. Right? I have the structure in 3D, but of course the structure is 2D. I have to convert it if I want to convert it to 3D. So I can say clean 3D, it gets converted to 3D. Of course it will be planar, right? So this is again an easy thing. Like if you want to delete, click select and delete. That's it. Installation, download, everything I have discussed in the YouTube video. Please watch that. I'm not going to take that time here because we don't have much time. Already it's 4. I have to finish within half an hour because I have another talk. So let us see the other results also. Now SDF, it has been read. We wanted to see whether it is read properly or not. Click the right button. At the bottom, you can see red molecules. You click it, you will be able to see whether the molecules are read or not. You will be able to see the content of the SDF file that is being read. Now, similarity fingerprint, click the right button. You want to see the similarity values, click that button. You will see a value here. So this is the similarity value. Between which and what, I will discuss in a moment. So there is a similarity value also being generated. Whatever the similarity value we just now saw, it is a comparison between the database and the structure I drew. Whatever the structure I drew is being compared with this database, and whatever the similarity values that you saw is that particular comparison. Okay. Distance matrix is something entirely different. Let me show you that. So I'm going at the bottom saying distance matrix measure. I think I didn't generate that. Okay, distance is generated, so it should come. Okay, let me take you to the viewer so that it will give you the right one. So I'm just viewing the heat map. Now you can see a heat map here. This does not say that this is your query molecule and database. You can do that, but you have to choose the right one. So I have to find out which one was my right one. Let me see. I am not able, I think I did not give the name. So whatever you see as the intercept line, that is the exact structures. Each row gives you each compounds. So if I mouse over them, let's say I am picking up this structure. So the compound ID 116, when it is compared with the row 1, the similarity is 0.65. If I come here, if the compound ID 152 is compared with row ID 102, then my similarity is 0.13. Let me go to the red ones. Red ones are higher. Anyway, you can see on the right side the legends. So compound row 11, say, so don't confuse. The row 119 is what is the name that I have given in the column. So if you give a proper name, it will come here. Whatever the numbers given as in the parenthesis, that is hash symbol 120, that is a row number, right? So the row 119 is what I have given as an ID, compound ID in my data set. So don't get confused. If you give a proper name, it will show up the proper name. If you want to see in a different color, you can do that colors. It all depends upon what kind of data you have to have a better visualization. And if you want to export this as a picture, you can do always export it as a picture. And let me see whether I can view the similarity to target. Yes, I can. So what here we do is, I'm selecting a specific compound. It could be from row 1 to 120. I don't know which one I can choose. I can choose row 186. It is trying to compare that particular compound with the rest of the molecules. That is what it is trying to do. So let's imagine row 81 is having 0.77, or let me, okay, it is automatically being arranged based upon the ranking. So row 81 is having 0.77. Let me choose another compound. Now you can see row 185 is having 0.62 percentage. This is really helpful when you're doing clustering or QSR analysis and all, so that you will get to know what is the role of your best active compound when compared with the other structures within the dataset. Another thing is, let me check, QSR model. I don't know whether I have run it. Let me try to run it. Okay, mostly it seems it's running, but some issue with aggregator. Let me check. Okay, predicted and predicted variance. That is correct. Okay, target column is not taken. That is a problem. So this one and this one. Okay. Now let me try giving the data. You can see it is running that. Now I'm running random forest. It is doing the prediction. Now it is running the meta nodes. Yeah, see, it is doing several times. See, iterations, they're doing several iterations. Statistics, does it do several iterations? No, because here it is self-learning themselves to optimize which model could be the best one. So it is self-training themselves. That is the difference mainly between statistics and there, if you want to do training, retrain it, you have to do manually. But here, the model itself, it is self-training themselves. So I have used a random forest. On the top, I have used a linear regression and also Gaussian process regression methods. It might take some time, but ultimately it will come up with the best model, whatever possible within the data set I have given. Let it do. I don't know how much time it will take. And each time you can make the model to write your model. So this is what I was telling you, deployment. So in the earlier picture, I did not show you model writing. So you can load this model also. Model can be loaded. So let me show you that mode. If I say model, where is model writer? Model reader is there, but that is for R. Let me check for the others. Yeah, it is, yeah, model reader. So if I read the model, I can apply the other data and it will be able to predict also. That is called deployment. That is also possible. Oh, I think now the model is done. Let us see the scatter plot. So this is a scatter plot. I will view the scatter plot. Okay, it is showing 1, but let me see. Oh, no, not 1. Okay, I have to give the predicted value. Yeah, this is the prediction. Okay, got it. So I can see R square is 0.49, which I got. This is all the values. Okay. So predicted with the predicted variance. So there is around 0.35 of variance is there, but between the experimental and the predicted value, I got around 0.49. And this, I can always check out what is the value here. It should pop up. And I can also give the range if I want to play around with the data variances or even with zooming in and zooming out. All those things can be done here. And let us see about the regression predictor, the predicted data. So this is the experimental value I have. I took only few compounds for sample data. I have this predicted value. And this data is not that great. Let me tell you, this data is for COVID, which I did some exercises on the activity data the experimental group has published. But yeah, 0.45 is what we get the maximum and not beyond that. And let me try with random forest predictor. What is the value they have given? So this is the experimental and this is the predicted. You can see it's a quite large. And then we also used Gaussian process also. You can see the trained model here. It telling you what is the average target value, lowest value, and highest value. That's what I said. Mostly machine learning, some of the methods are more of black box, not open box. Until unless you predict them, you don't know what it is happening. Yeah, this is another one where you loaded the SDF. You can predict multiple properties. Let me show you how those properties. So yeah, I was also telling you that I will show you the different fingerprints. Ardicate fingerprints. Yes, Ardicate fingerprints. I'm just connecting this. Without connecting, it will not show. When I double click here, you can see what are the fingerprint types I have. Morgan, feed Morgan, atom pair, torsion, ardicate, avalone, layered, max, and pattern. Of course, daylight you won't be having. But daylight, I think CDK is using daylight. Let me check that. CDK. Yes, fingerprints and CDK. So some nodes has some requirements. So read the README and then that's how you have to take it. So here you have standard extended E-state, PubChem, Max, and circular. Here also ECFP is also there, but here also we don't find daylight. So yeah, I think being it's paid, probably you might not find it anywhere. But I think Marvin is having. Anyway, that is, we could check later. So here, let us see what are the properties we can get. So when you double click it, you will see these are the properties. These are all chemical properties or physiochemical properties, nothing like ADME properties. And then random descriptor like S log P, TPSA, Lipinski rule, number of atoms. Then let us see other molecular properties. Most of them will be repeating. So I'll show you how we have done pruning the data. And NLOs, as I told you, we need to have exe file. It works only for Windows. So I cannot, I'm in Mac. Then your PADL descriptors anyway there. So all your PADL descriptors will be, could be predicted here within KNIME. That is also possible. And when I'm joining, I'm defining that match all the following. So I'm saying match the row ID with row ID, and the column selection, I'm selecting the column, and I'm also saying remove joining columns from bottom input. That means if it is any duplicates are there, remove them. And append the suffix if there is any duplicate column. If I don't want to remove it just to do data analysis, because I have to first do some data analysis and then only I have to simply remove. But exact copies anyway it will remove. If the correlation is a bit closer, then I have to take much care. If I want to view the, so duplicate row filter is also there. If I double click on that, it will look at all the rows, and then it will duplicate, it will check it, and it will try to remove the duplicate rows also, and it will try to keep the first one if there is a duplicate. And then I can write it as a CSV file. But if you want to view them, let me see. Oh no, I think I cannot view. It is already written. That's why I cannot view it. Now, yeah, this is another thing for Open Babel. If you want to convert files, so I have SDF file. I'll go to Open Babel. Keep in mind, Open Babel works seamlessly with Linux and Windows, but for Mac, you should have your Open Babel installed separately. I can convert any format from two other formats, because whatever Open Babel supports, it supports. Here I can convert from SDF to 3D. And if you want to generate a 3D, of course, you can give that optional parameter saying that gen 3D, it generates the 3D structures also. So both ways it is possible. And then, this is what I was telling you about cleaning your data set. So I read SDF, I can read the molecule. I am removing the salt forms. Most of the time QSA does not work for salt forms and also for docking. So I want to keep the largest fragment and remove the other fragment. I'm adding hydrogens. I wanted to optimize my structure. If you double click, you will see, I think, okay, you can see which force field to be used for conformation generation, or sorry, optimization. And then you can say, you can give a new name for the optimization, and you can also give how many iterations you require. If you have a largest data set, then give at least 3000. Smaller, 1000 is fair enough. And if you already have a 3D structure, but you wanted to generate a 2D structure, that is also possible. So you give the optimized to 3D structure, and you can generate a 2D structure. And let me see, conformers. So this is, you can draw the structure. I have already drawn a structure using Chem Sketch, Structure Sketcher. This is not Marvin, this is another JChem paint. And then I used RDKit and I generated conformations. Let us see the conform. It will take some time, so I have already ran it, so I have the results ready with me. So these are the structure generator, but let me see the energies, because I asked it to generate only one. So force fields, yeah, here I can see the energy values in MMF and UFF. Similarly, I can do a template docking, template conformation. Of course, there might be plenty of them, so that's why it is taking time. Let us wait. But yeah, feel free to ask questions now. Anyway, yeah, you can start asking questions if you have, or if you want to put it in the Menti, also we can start the same link. I hope Parshuram sir has already sent you the links for the Menti. You can in the Q&A, you can press the click that button, and you can put your question if you want to ask anonymously. But if you are free to ask, of course, you can unmute yourself. I think because of a lot of data, it is taking a lot of time to load. That is absolutely fine. Then another one is clustering, props, model, HD, yeah, SIPs I can show you. So this is also, I showed you in the slide to show you which particular isoform of the P450 it is being showing. So it will show you the which predictions. Oh, this is also taking time. I think there is too much of data, and I have already opened a lot of applications. That could be the reason. Otherwise, I'll try to run it again and see. Oh, somewhere it is still running. That's why. Okay. Let me check where the job is still running. Is it? Okay. Some model. No, it's all done. Because it seems like in the SIP okay, but I don't know why I'm not able to see the data. Okay, now it came. So here it tells you that this particular atom has a selectivity to that particular isoforms of the SIP P450. It doesn't give any value, but it just tells you about the P. So let me show you that column. So here we have 2C9, 2D6. These are the columns actually. So each column is for each isoform, and it is telling you which atom is sensitive to those particular isoforms of the P450. Anything else? I think mostly I covered. Yeah, mostly I covered. And finally, moving forward to some of the codes. So this is where I have used a lot of Python wrappers, nothing to do with machine learning, but a lot of Python wrappers are being used in these codes. I have a modeler also. No, modeler is in the other one. I'll show you that. Yeah, here. So many of the time, my students used to tell me, so it's very difficult because the script might work, might not work, and we are not able to see the results. It's a bit not of interest. So is there any solution that you can make everything on a single page so I can see the script, I can see its corresponding output also, so everything will be easy. That is absolutely true. And I just tried with Gromax and Modeler. It really worked, and I shared with the developers also. It will be soon there, will be with them. They'll be referring to these documentation. So it will be easy for students to learn. So what we have done is a few here and there tweaks on the tutorial, but this tutorial, even though I have used the latest data. So not the old data. Let me refresh it, because even when you load also the GitHub will be, because it has more lines, so it will take some time. So you have to refresh several times, it will come up. So you can see here, I have the script here, the Python script here for protein modeling. So the first script is actually saying that to do a sequence similarity search of your sequence against the PDB database. Of course, you have I have given a link to download the PDB database, which is just a 60 MB, 60 MB. So don't think that it will be GB. And after doing the similarity analysis, they have generated a table to show the sequence analysis. It also told you e-values, and then we were able to generate the profile, build the profile. Okay, this is all output. Okay, so the input and output, everything will be in the single page. That is a benefit here. And this is again aligning target sequence with the template. And then you can do result analysis and finally generate a graph. Here also I have used a matplot, lib or pylab. So I'm just comparing with how far my residues are being well modeled when compared with the template and with the sequence that have been taken. So this is already published in Senado. You can always use it. Similarly, I have done this for Gromax, whoever works for MD simulation. Now, many of you might think about RDKit. I purposefully avoided showing you RDKit in Python. There are plenty of tutorials available. Why should I repeat the same thing again? So